Okay, so it's my pleasure to introduce Dr. Gary Miller. Dr. Miller is the Vice Dean for Research and Strategy and Innovation at Columbia University. He also is a professor of environmental health sciences and professor of molecular pharmacology and therapeutics at Columbia University here in New York City. He's also the director of the Center for Innovative Exposomics, and today he will tell us much more about the exposome. He has been a leader in Exposomics, which studies the comprehensive and cumulative exposure to physical, chemical, biological, nutritional, and psychosocial mediators that impact biological systems. I first met Dr. Miller when I attended the Columbia Summer Boot Camp in 2019. And I got really excited about the concept of the exposome. As you know, our department is interdisciplinary with multiple expertise from GIS to environmental health, to nutrition exposure, to water exposure, and epidemiology, nutritional epidemiology, and environmental epidemiology. And at the time, we were looking for a concept that glue us all together as a department and works collaboratively. So the concept exposome was quite fitting as we were developing our new PhD program in environmental and planetary health. And that was a concept that will have all our expertise in one area and looking at different areas of the exposome, both the external exposome and the internal exposome. So that was fast forward. I also attended again the Columbia Boot Camp in 2023. And just those 5 years have passed with the advances in the field of exposomics, which Dr. Miller will discuss with us today. Also, Dr. Miller had helped to develop this high-resolution mass spectrometry techniques and methods to provide an omic scale analysis of the human exposome. So in a manner similar to the genome, the epigenome, the metabolome, the transcriptome, and the microbiome. He also serves on the advisory panel to the National Institutes of Health, All of Us Research Program, and the National Environmental Health Sciences Advisory Council. He's also the founding editor of the journal Exposome and has previously served as the editor of Toxicological Sciences. So please join me in welcoming Dr. Miller, and we really appreciate your coming to meet with us today. Thank you.
Thank you, Gada. I really appreciate the invitation to be here. I moved to New York about 6 years ago, and this is my first visit to CUNY. Like, I've had so many interactions with people, but actually the first time being here. We did have a pandemic in the middle of that too, which caused some challenges. So yeah, I want to spend some time talking about this concept of the exposome and the work we've been doing on this for the last probably about 12 years or so. Let's see, will this work or not? Okay, so if I were to show you a brief history of genomics, you would go back into the late 1800s and things and looking at inheritance. Exposomics is relatively young. Chris Wilde coined the term exposome in 2005. Shortly thereafter, he became director of IARC. So he was a cancer epidemiologist that was really interested in finding an environmental complement to the human genome, because the Human Genome Project had just finished up about that time. And so he introduced this concept, and for 5 years, there was not another paper that mentioned the word. And it wasn't until he wrote a second paper that there was the second paper that came out. But then about in 2010, there was a workshop at the National Academy of Sciences to kind of explore this idea. And that was really when things started moving from my view of this. It wasn't until after that meeting I actually heard about this. One of my friends attended that meeting and came back and told me some about it. Another important finding was in about the same time, 2010, Chirag Patel, who was a graduate student at Stanford, published an environmental-wide association study for diabetes. And that was like that first attempt to get to that genome-wide association level study of an environmental disease. And so he published that work again as a grad student. He's now a faculty member at Harvard, and he's someone I've worked with quite a bit over the years on this. Then about in 2013, the European Union funded a few Exposome grants. They were very interested in this area. And about that time, the Hercules Center, this was when I was at Emory University, was funded as an NIEHS P30. And when we developed that center, the thinking was that the exposome was a really interesting concept, but we didn't really have the tools to measure it. And so we developed the center with the idea of like building the infrastructure to be able to do it. We weren't ready to do it, but we knew we had some tools to build. And so we spent several years doing that over the years. And then, you know, in the last 5 to 7 years, there have been many more initiatives, again, often led by the European Union, many different activities and things going on. But now, as we've seen there the last 2 or 3 years, there's been a large uptick in interest and funding from multiple institutes. And so this concept, again, Chris Wild's view was a sort of complement to the genome, trying to capture throughout our life course all the things that we're exposed to. And I think my background's in toxicology, and so I would typically think classically about the sort of things on the physical chemical side. But it was very clear when you're thinking about all the non-genetic drivers of disease, it's much more than that. And so we're thinking about the sort of social determinants, lifestyle, ecosystem things like green space, blue space, all the way down to socioeconomic things. But to me, one of the key parts of this was that for any of these external forces to have an impact on our health, they have to affect our biology. Could be indirect, but the idea is that if there's something in your surroundings that's making you sick, it's not magic. There's biochemistry involved in that. Could be epigenetic sort of signatures, whatever it is, there's a connection there. And so to me, a big part of the exposome was linking it to those biological consequences, not just describing them from an association standpoint. And so one of the challenges here was that over the years, people interpreted the exposome in different ways. They referred to different aspects of it. They might be focusing on like the external exposome versus the internal, or the social exposome or the chemical exposome. And to me, like, I always understood like where people were coming at from their angle, but I kept thinking, if you're trying to explain this to someone like Francis Collins, the director of NIH, having a bunch of versions of the explanation of a new thing was not very helpful. And so this is something we struggled with for a while as different people were putting out different versions of their interpretation of it. And so about a year and a half ago, I was working with Tom Aniotis, who was directing the Precision Medicine Institute or initiative at Columbia. And he said, you know, if you're struggling with something like a definition for a field, he's like, you should hold a meeting at Cold Spring Harbor. This was done a lot through genomics. He said, if you had a Banbury conference, you lock people away for like 3 days in a room in this isolated area with a goal of achieving something. And so that's what we did. So we held a Banbury conference on integrating exposomics into the biomedical enterprise. And a lot of my thinking was like, what is the best way to define this from an NIH perspective? We knew we had a new NIH director coming in. There'd been, you know, turnover there. It's like, how do we really explain to people what this is? And so this definition is what we arrived at that meeting. We had a few people from Europe attending this meeting, and they've introduced it on their European exposome groups, and they wanted to add a word or two to it, which we're still kind of grappling with. But the idea is it's this integrated compilation of those physical, chemical, biological, and psychosocial or social determinants of health and disease, and even the biology part of it. And so we submitted this to a journal, and it sat on the editor's desk for almost 3 months. It was quite painful. We got the reviews back just a few days ago, but it's, I think we're now getting close to getting this published in a high-level journal. But it took a lot of work to get to this, like to come up to a kind of a consensus definition that really was trying to be all-encompassing and not leaving people out. And then again, at the meeting, we had several other sort of like objectives and directions the field had to go in. But it's something where the goal was to have something where, an NIH Institute director or a CDC lab group or something, you can say, this is what we mean by the exposome. Because this was something Rick Weidchick, who's the director of NIHS, said he was always asked about this. And when he talked to Francis Collins about it, he's like, well, how do you do this? How do you do an exposome experiment? But you have to know what you're talking about first. And so this was a lot of struggling to get through in this point. And I think that these sorts of meetings are necessary to move a field forward, complemented with like larger, more stakeholders involved, larger size meetings. And we have some of those planned. But one thing that we were in Brno, Czech Republic, a couple of weeks ago with the NIHS director, and we decided to plan this meeting for next October to see if we can get the exposomics and genomics communities together. And the idea is we're going to hold the meeting at the Mendel Museum, which is where Gregor Mendel did all his pea experiments. And you can actually walk out on the lawn, which was where his garden was, where he actually grew the peas. And you can have pea soup at the restaurants nearby too. But it gets back to Chris Wild's idea of complementing the genome with the exposome. And so the idea is like, can we get the director of the National Human Genome Research Institute, from Wellcome Sanger, Broad Institute sort of things, to really get the key genomics leaders in with the exposomics leaders to see how we can get to this sort of G by E understanding. So there's a lot of these efforts to kind of help, drive a field forward. It's not just doing the research and publishing papers. There's a lot of things you have to do at a scientific community level to get these things moving along. So from my perspective of this is that I'll come back, I really value what we can get at like geospatial information and, social factors into health. But my more laboratory-based research, what I saw as a great opportunity was to use high-resolution mass spectrometry to measure what we can measure in biological samples. Like we know from metabolomics, we can measure all sorts of things in the small molecule space. And a lot of the people that have been studying metabolomics for the last couple decades, if they saw a phthalate or a flame retardant, that was like noise to them. It was contamination. It wasn't something part of that individual's biology. Whereas we saw it as like the same sort of mass specs you're using for that are actually picking up these peaks that correspond to pesticides and flame retardants and changes in biological responses from that. And a big part of this was that over the past 10 years, the mass spectrometers have just advanced so much. Like you think about how science advanced when our microscopy got more accurate. You went from, Leeuwenhoek looking at a cell, and now what we can do with cryo-EM and getting angstrom level structure is as you improve your resolution, you always learn more. And so in the olden days, you would see a peak on the mass spec, and there could actually be like 10 different things inside that peak. The new high resolution instruments really spread these things out. So now we actually have much better resolution of these peaks. And so when we see 100,000 peaks on something, we know they're all individual peaks. We may not know what all of them are, but at least we can now separate them in that way to start doing that interrogation. And so this is really what, when I was at Emory, Dean Jones was the one that really pioneered a lot of this work of like using the mass spectrometry, not just to measure what's in our bodies naturally, but all the external factors that are in there. And what we found was that you really have to use more than one method to do this. And so we would do liquid chromatography, high resolution mass spec with different types of columns, trying to separate the different molecules, but found that it was hard to get a lot of the coverage of the semi-volatiles and more environmental chemicals with those methods. But the gas chromatography, high resolution instruments started coming on board about 7, 8 years ago. And we said, well, if we did it on both of those, now you get really good coverage of chemical space. You get these more, non-polar molecules of the GC and the LC. You combine them together, you get this really good coverage of chemical space. And so we developed a lot of workflows. There's a lot of data processing, a lot of informatics and modeling and things to do with that. But the idea is that we can detect hundreds of thousands of peaks, and they're identified with varying levels of confidence. It's not like we know exactly what all of them are, but when you have a mass to charge ratio out to 4 decimal points, you have a lot of good information about it. And like a lot of people like to refer to these as unknowns, which is fine, but they're not unknowable. They just need more work to figure those things out. And so this is what we formed this new center just over the last year. And as we were building things out at Columbia and more people were coming to us asking to collaborate and help with things, is that it was getting more than like my own lab could do. And so we formed this center that's really focusing on building out these technologies and help with the training and methodology and promotion of it. Since I arrived, we actually have managed to have, we now have 6 of these high resolution instruments, combination of a lot of grant funding and things, but really set up this lab to focus on doing this sort of exposome level analysis. And again, we initially built this based on a lot of the model we had at Emory. And one of the points too is that can another lab replicate what was being done in Atlanta? And so this has been a big part, is it's like you have all these different labs doing different things. If you can't do some harmonization, it's hard to believe some of the science. And so in thinking about like how we identify these things, in metabolomics, we use this Szymanski scale of like, level 1 confidence. You have a reference standard for it. You know exactly what it is. All the analytical chemists are happy. They would know it's there. And I like to kind of imagine if you're trying to describe a car, that level 1 is like having that vehicle identification number. There's only one car like it in the world. You know exactly what it is. And that's great if you have a reference standard for it. But if you ingested some food that had a pesticide residue on it, and that pesticide got metabolized by some bacteria in your gut and then got into your body, you don't have a reference standard for that. You have some unknown metabolite that no one knows what it is. And if you're going to get a level 1 or 2, it's just not going to happen for that sort of molecule. But that molecule could be a carcinogen or something, so it could be very important. And I think what we do is like we get this, exact mass with these level 4 and 5. We really need to get more in this sort of level 3 part. But imagine like if you had a license plate number of a car, it's enough to find that car and repeatedly find it again. Even a partial license plate, if you knew the model and things of it. And so the idea is that you don't always have to know that VIN number. You don't always have to get to this level 1, is that it's okay to be good enough. Because if I see this peak showing up in a study where I'm looking at asthma in one population, and then I see it and I replicate it in another study, then I can say, okay, I saw that peak coming out at 12 minutes with this mass to charge. I can now go use other techniques to figure out what that peak is. But I'm not going to spend time identifying every single peak until I know it has an association with a key outcome. And so this is an example of, again, like these sort of coordinating efforts we did. My postdoc, Yoon July, led this, is we had colleagues at 20 different institutions from 7 different countries talking about how to use these approaches to measure the exposome. Because again, we have different labs doing different things, like just thinking like, where are these sort of gaps in the field where we have to do? And again, like getting people to agree on some of these key issues, it can take a lot of work to do that. But it really starts getting out what are some of these challenges, what are the needs in the field, and how do we get to that? So one of the earlier examples, this was something we started about 8 or 9 years ago. It was an NIH Common Fund project on chemical identification. And what came from this was that just like this simple observation that these coumarins in grapefruit juice we know inhibit cytochrome P450s. And they can disturb a lot of different drug metabolism. If you think that grapefruit juice is the only thing in our environment that alters metabolism, you're crazy. There's many other things that do that, but we don't systematically study them. And so one of the things we started talking about was that could we do a better job of identifying what some of these xenobiotics are that do change how we metabolize drugs? And so one of these projects we did was we looked at generating some of these metabolites. So again, if you don't have a reference standard, it can be hard to identify something. And so the thinking was, if you had a 96-well tray and you put liver fractions in it, and you put a known chemical into one of the wells, and then sampled it every few hours, things that showed up new that weren't there at time 0 are metabolites of what you put in. And so essentially you can develop like a pseudo standard library saying, yeah, we know this is a metabolite of this parent compound. And if you're doing it at the sort of 96 well trace standpoint, you can start doing this for hundreds of different molecules. And so Dean Jones' lab set this up. We did this in collaboration where you could go through and because you have liver fractions, you're getting phase 1 and phase 2 metabolism, and you generate a lot of different metabolites. And from this, like we can see that over time, we see these things generated. We can actually then take those parent compounds, inject them into a mouse, and take samples from them over time. And so if you're seeing things that matched up from what you did in vitro to what you saw in an animal, you're like, yeah, we know that that's a metabolite of that thing because we just saw it and measured it. And on the far left is actually looking at human samples. I think that's right. No, this one here. This is the one that is the human samples where we were looking at metabolites of a drug like bupropion, and you identify these novel metabolites that show up in the people that are taking the drug, but not in people that aren't. So the idea is you could take this from this liver fraction to mice all the way to humans. And during this work, one of the postdocs said, I think I found a novel metabolite of caffeine. I'm thinking, people have been studying caffeine for decades, so you know everything about it. But it turned out it was. There was this metabolite looked like caffeine, so we went back and did like a isotopic labeling of it and showed, sure enough, this was a metabolite of caffeine never reported in the literature. But now it's reported in the databases, so it could be found by someone else in the future. And so our thinking was like, could we do more of that? And I'll come back to this. One of our earlier studies, really kind of pulling together these tools with the LC and the GC, started probably about 7 or 8 years ago. A group from the Mayo Clinic reached out to us. They were studying primary sclerosing cholangitis, liver disease, and they had heard about the exposome and didn't really know what it was. This Costas picked up the phone and called me. And we said, we just got this GC mass spec and we could do GC and LC on these samples. They had very well curated samples. And essentially what you're able to do is like do a network analysis of how certain chemicals that you see detected are corresponding to changes in biology by doing like a network analysis of the biological pathways to those environmental chemicals. There's many chemicals we detect that don't seem to have any sort of correlation to it. But this showed that we could start using this as a discovery tool for a range of different conditions. What was also interesting to this was that shortly after we started this collaboration, the Mayo Clinic was awarded the biobank for all of us. So we were getting the samples from the Mayo Clinic biobank, and then it was then scaled up to the all of us one. And so we've always been kind of thinking about what would it take to go to that sort of level and really scale those things up. And so some like the examples of this, the way we do these studies is that when I first arrived here, I was talking to Richard Mayhew, who leads the YCAP study, which is the Washington Heights and Inwood Community Aging Project, which is really focused on Alzheimer's disease. And so he said, he's like, well, yeah, we can give you different samples from this. And we just threw it on our first instrument, the LC we had at the beginning. And you could easily separate cases and controls from that without a problem. But we also saw separations of the different racial ethnic groups. So Blacks, Hispanics, and whites separated. And he was like, how can that be? He's like, there's no genetic differences. We've never seen any other difference among these populations in all the work we've been doing. And I said, well, it could be something as simple as diet. And he said, that can't be because we did a dietary inventory. I didn't see any differences. And I said, you know, Richard, that's not biochemistry, right? Like you're asking someone to record something, you're not actually measuring what's there. And what's really great about this is besides measuring those molecules, you're looking at the endogenous biology. And what we saw was that the biological pathways they were different in the different racial ethnic groups that have differential vulnerability to the disease. And so it starts connecting you to like, why are these populations more vulnerable at a biochemical level? And so we did this initial study with just like 160 samples, but then used that as preliminary data grant for like 3,000. And so we have like samples from over 3,000 people, multiple time points, longitudinal, we have incident and cross-sectional design. It's a really amazing study. And all these things are in process, and it's been very exciting to work on these things. Again, in a very diverse cohort that's very well characterized from a phenotypic standpoint. One of the observations was the Caribbean Hispanic population in Washington Heights seemed to have a higher incidence of Alzheimer's. And so Richard asked the question is like, why is that? Like, what's going on with that? So he started a study in the Dominican Republic and recruited patients, was actually able to get cerebral spinal fluid from several hundred of them and plasma. And we were able to go through and like do this sort of comparison. And during this time, some protein biomarkers of Alzheimer's had been developed, these phosphorylated tau molecules. And so we added that part into it where they could say, what if we look at people who have a clinical diagnosis of Alzheimer's versus those that have that clinical diagnosis, but also those elevations in the protein biomarkers that are thought to be that biological basis. And what you found was that these are different groups. So almost 20% of the people in the study that were diagnosed by very good neurologists didn't have the protein biomarkers of the disease. They had dementia, there was no question about that, but it's a different subtype of the disease, right? It's a different thing. And if you're trying to compare groups at a molecular level and you have 20% misclassification, like you really lose a lot of signal. And so when we started doing this, you just get very different results when you can classify it with a biological marker of that disease. And so I think this is important for multiple conditions. And I think, for example, something like autism, it's probably multiple diseases. And if you're studying them all together, you'll never figure out the different biology. Like we need to have other molecular markers or better phenotypic markers that can stratify these conditions across what is the reality of that continuum. And so one of the things I'm really excited by this is that you can take those individual biomarkers like that p-tau181 and using the mass spectrometry, essentially do an environmental-wide association study against that molecule, not just the disease itself. So the idea is like what in the body is changing in corresponding to these protein biomarkers, to think about what are the markers of progression of the disease or pathways that are being disrupted that could be targeted for therapeutics. So it really gives you this very open way of looking at what is all the change in biology in these populations in this way. And so we've been, again, doing a lot of these things, doing a lot of analysis. Some of the papers are starting to come out, but it's been very exciting to have multiple cohorts to draw upon to do some of these studies. And so this was, I think this one, I have like two geospatial type slides here for you guys, is in that initial study, we asked Richard if he had residential history on the people. And he's like, yeah, it's all geocoded. We have all that. And we know we have a lot of data in New York City about air quality and other things. And so what my graduate student at the time did was linked all the different air pollution measures to all these small metabolites we had seen in this population. And looking at black carbon, PM10, PM2.5, et cetera, and what she found was that the PM2.5 pulled out glutathione metabolites, the cysteinoglycines, were elevated in the people that had the higher levels of PM2.5. And so what that to me was like really striking is you have these very macro level measures of this external environment linking to these biological changes that could be linked to some of the biology involved in the disease process. But all we were looking at was she just had air pollution data. But there's so much other data out there about that's coded across the United States and the world, right? We have information on all these different factors from green space and blue space, meteorological measurements, our light at night, noise factors, walkability, there's a lot of social demographic sort of things. And so if you have the geospatial data, there's this opportunity to link in these biological changes to all these more macro level parts of it. So, but you have to have the data in that right configuration to do that, to link these things together. But this is something that we think is going to be very important for the field because we can link these very diverse data streams together. Another example of a study we did, I've been working on Parkinson's for about 30 years, and it was really one of my motivations for starting to work on the Exposome was to be able to come back and do this for Parkinson's disease. And so we did our first study last year where we got several hundred cases from the European cancer study EPIC. So they enrolled the people back in the 90s. And then over the intervening years, a bunch of them developed Parkinson's, had incidence PD. But we got these 25-year-old blood samples that were taken about 10 years prior to diagnosis. So there was no reverse causation going on here because they didn't even know they had Parkinson's disease. We do know that caffeine consumption is protective in Parkinson's. And so one of the first things the group did, they said, well, like, what do we see with the sort of caffeine consumption? So why don't we measure caffeine and its metabolites just to see if it corresponds to what the self-report was of caffeine? Like, as we know, some self-reporting is good, like cigarette smoke and coffee consumption. People are pretty good at recalling what they do. And that's exactly what we saw, is we saw the caffeine metabolites we were measuring matched up to what they had reported in this from years ago. And so, but what we didn't do is we didn't just measure caffeine in their blood. We measured all the caffeine metabolites there are. And so you're picking up like a pathway level analysis of this one environmental factor involved in the disease, even though it's protective. And that bottom one there, the trimethyl dihydrouric acid, is the novel metabolite we discovered the year before. So it's like we actually found this thing in a human population from 25 years ago because we put it in the database. And so now this was like, we were doing this very much in a targeted way just to look at these metabolites, but we have this full non-targeted analysis that we're working on right now. We've run it through the mass spec. It's just the data processing takes a long time to do all these things. But it's very nice to have this population well before diagnosis to see what things may be contributing to the onset of the disease. As a laboratory person, like I like to kind of get in and think about controlling all these variables, and had done a lot of mouse work in my career, but it just wasn't the right throughput for doing exposomics. And so we adopted C. elegans in the lab about 8 or 9 years ago, and now use this as a way to kind of follow up some of these connections. And so when we saw elevation of an insecticide in Alzheimer's disease, a DDT metabolite, I had my student like get C. elegans worms that had the genetic mutation for tau, like the humanized version of it, and treat them with that pesticide and do a kind of G/E analysis. And what's amazing is that all the mass spectrometry I talk about doing on human blood works in worms too. So we're doing this comparison, and she was able to show that biological changes in the DDT tau worms, there was overlap with what we saw from the cerebral spinal fluid of 80 patients. Like there was that much conservation in biology between that. And so the idea is like, it's a good way to go back in and look at some of these G/E sort of observations you have to see if you can kind of get down to these sort of mechanistic toxicological connections to it. So it's something we've also been doing that for a while, and it's a very nice way to kind of bring some biological insight to what we see in the human studies. Another study that we just started a few months ago is this project called SAMBI. This was a Cancer Grand Challenge grant. Melissa Davis at Morehouse is the PI of it. She had been up here in New York at Cornell before. But what's really exciting about this project is that it's covering the social determinants of health, exposomics, genomics, and tumor biology. So I'm leading the work package on exposomics, but we have very detailed social epidemiology measures, like very long survey things that Lauren McCullough at Emory is leading. But the goal is to enroll about 40,000 participants, and they'll get all that social determinant data. They'll get all the genomics. We'll do the exposomics, have the tumor biology, and can start linking these things together to figure out why are there disparate outcomes in cancer. And so specifically what her motivation here was looking at the African diaspora, like why do you see these higher incidents? And so we have 5 countries in Africa and also Blacks in the UK and US to see if they can tease out these differences in the various exposures and things. So again, using that workflow we had for that. But this just started a few months ago, and we're very excited to be able to contribute to this project. And again, this is the same sort of thing. We're just throwing all these things that we do across all this high resolution mass spectrometry. And one of the goals too is actually trying to get some of these mass spec approaches that we're doing here exported to the lab in South Africa, because they do have some of the TOF instruments, but we need to kind of deliver them an exposome assay. And so we're working on validating and developing that as well, so that they'll be able to do this on their own even after the project ends. And this is like an example, a pilot study we did with Mary Beth Terry at Columbia using these sorts of approaches this is a case control design for breast cancer. And so on this side, you're seeing the things that are elevated in the population, but things like dibutyl nitrosourea were elevated. So these things that are known carcinogens, we see elevated in this population in a pilot study. Like it's like, I don't want to put too much stake in that molecule, but it's not just chance that these things come up like that when you're looking in these sorts of designs. But as we know from genomic studies is that you do need to have higher power to get to some of these nuances. And that's what this very large study will bring us. Like this was just about, I think, 150 patients or something in this one. But again, we found many different molecules that were elevated in the people with cancer. And again, kind of the same thing, like we're able to do these sort of Manhattan plots to look at this small molecule space for any sort of condition or disease, whether it be just the diagnosis or some phenotypic measure you're looking at for that. And so we're doing another study with them where they've done like breast imaging and looking at changes in those images compared to the sort of environmental exposures before they even develop cancer. So it's just really about what sort of study designs you can draw upon to look at some of these connections. Another recent project we just got funded that's been very exciting, kind of following up on this idea of the drug metabolism piece. As we started talking about that, and I was talking to the director of the All of Us program, which will have 1 million people enrolled in a couple years. It's a large biobank, so they have samples from all those people in the biobank. And we were talking about exposomics, and I said, well, you realize that a lot of these environmental factors are disrupting how people's medications work. 20% of people in the US take 5 or more drugs. We don't study the drug combinations. Like it's the clinical trials look at one drug and they get it on the market and they go. And we said, well, our mass spec is getting pretty good now. If we put some effort into this, we could probably measure the top 100 FDA approved drugs and all their metabolites and thousands of chemicals that interfere with how they work. It just will take a lot of time and money to do this, to scale it up in that way. And so that's what we pitched to ARPA-H. ARPA-H is like that new DARPA-like funding agency in the US, looking for like high risk activity that NIH can't do sort of things. And so what we're doing with this is one, using the Odyssey electronic health record database. If you're not familiar that George Ripchak is the lead on the Statistical Coordinating Center, but he has access to electronic health records of 12% of the world's population, like 1 billion people. And so if you're trying to see like, well, what drugs don't work as well as they should? Like where do you see interaction effects or more side effects? And so he's been mining that data to kind of guide us and to see where are we seeing the biggest gaps in this, and also to provide like some real world validation of the other things we find. We're working on generating all these standards of these approved drugs, the environmental modifiers, and even like the inert ingredients and the binders and medications that again don't get measured in a systematic way. And then together with the Mayo Clinic and several clinical trials, we'll be doing this very in-depth analysis of over 10,000 patients who will also have whole genome sequencing on, and just trying to do this very, very deep dive to measure all the drugs that are in their system, all of these environmental chemicals, and then drive this towards like actually getting CLIA approved tests for this. So the thinking would be for some conditions and some medications, you would develop a test to say if you're going to go on a mood stabilizer or something like an antidepressant, is have an assay that can measure all those drugs and their metabolites and the things we know interfere with it to complement the pharmacogenomics. So you can do the pharmacogenomics and the pharmacoexposomics at the same time. And then really work on scaling these things up in a pipeline format so you could get to the sort of biobanking level. It's like my goal is to be able to do this at a scale of a couple 100,000 samples. And while that might seem kind of crazy, if I can get a grant now from NIH to do 10,000 samples in 2 years, if I just get more mass specs, I can quadruple that, right? I mean, you can just hammer the approach, but if you can get the speed better and more automation, you can actually get to a level where you could do that in a systematic way. And so again, it's multiple collaborators here, multiple universities, partnerships with Thermo Fisher and AWS, with the big goal of like getting these things to a product level in that way. And so This just started a few weeks ago. Very excited to do this. It's a big, I would almost say it's as much of an engineering project as a biology project, because it's really a lot of this scaling of things. And again, this, for an example here, is that you look at these different cytochrome P450s, other enzymes, we know that there's many different factors that can affect them. We know there's genetic variants that can affect them. And there's this whole range of drugs that we know that they metabolize. But no one's ever systematically tried to assess this. It's always kind of like one at a time sort of analysis. So this is like the goal of like being able to fill these things out using all these different approaches we've pulled together. The next project I want to talk about is, for many years, I've been harping on the need to have more harmonization of our approaches. Again, we have all these different labs doing exposomics, all in their different ways. And that's not how you convince other scientists that it's a real thing. And so NIEHS, along with the National Cancer Institute, National Institute of Aging, a couple other institutes, put out this call for a coordination center for exposomics. And Shrag Patel at Harvard, Rima Habre at University of Southern California does a lot of geospatial work. And I put in this application with many others, which I'll show in a second. But the idea was to really promote these national NIH level, but also more global initiatives just to start getting more of this coordination across this field. We have cores for chem, biological, analytical sciences, geospatial sciences, and data sciences. And the idea is to really kind of start building out what this field will be. And so we have these different groups, and there's leadership in them, but you'll see they spread out quite a bit. But the idea is to really kind of set the standards for the field, thinking about the right protocols, harmonization, educational sort of opportunities as much there as possible. So there's a lot of people involved in the leadership team. You'll see Gada down there. And one of our key outreach pieces, we want to make sure that we're getting the tools delivered to the people that need them, and the things that we develop are available, in an equitable fashion across the world. And so we have these different groups. We have these collaborative hubs that help push these things out. Multiple people from multiple universities, very complicated network to do this. But I think we'll see a lot of really strong programs come out of this and get much better coordination at the NIH level. I was talking to God about the boot camp that we've taught every year. This is something we were doing at Columbia, at Mailman. It's a very expensive course because it's like funded by our department in environmental health sciences. But the nice thing about the Nexus grant is we have the NIH support. So the courses we develop out of this will be much more affordable, much easier to get onto and stuff, because we want more people to be able to have access to it. Again, we'll continue to build out the sort of educational pieces of it with many different types of interactive workshops, which we're developing, and really just kind of trying to continue to expand the network from what we've done at the boot camp at Columbia, and really just trying to scale this out at many different levels so people can learn as much about this as possible. We've been working really hard and just promoting the concept, just thinking about social media and getting things more in this range. A lot of really great engagements with industry, from the people that make the instruments to like Google Earth, Amazon Web Services. And we've had multiple conversations with venture capital groups, not just in New York, but also in Boston and California, that they see exposomics as a growth area. There's a place for companies in this space, which we knew was really critical, like with the Human Genome Project, is if you don't have those partners to develop new tools for you, things don't move as fast as you'd like. Another great exercise we're going through on the analytical side was we want to do like an exposome ring trial. We want to get samples, send them out to 20, 30 different labs, and have people throw their best techniques at it to see what they can detect in those samples, and then compare across those different groups and technologies. So many of them are using, GC or LC, NMR sort of things. We had 24 labs agreeing on the application when it went in, but when I was in Europe a couple of weeks ago, like 10 other labs said, yeah, we want to be part of it too. So people want to really see what they can measure with that. And I think, one of the goals of this is really coming up with like an exposome kit. So you don't have to have high resolution mass spectrometry. You develop tools that can be used in a much more inexpensive way to do these for people that don't have access to some of this instrumentation. And so this is something that's underway, and ultimately, I think it'll really help advance where we're going in the field. And then I said Rima Habre and Raja Sakur at UNC are leading the Geospatial Hub. Rima helped host this workshop at the National Academies about the sort of wearable technologies, and just thinking about what are all the infrastructures you need to scale these things up. Like, how do you get very good workflows and the best sort of systems to do this? Crystal Pollitt's group at Yale, who's involved with our analytical core, is the one that developed that fresh air sensor, where it's like, it's kind of worn inside a watch, so you don't have like touching and skin contact to it. And so really trying to bring the best tools in, having the partnerships with groups that are doing this. I think there's so much potential here. I've seen other data about like activity monitors and how important it is for, like, you can alter people's behaviors in positive ways, and you have ways of recording that. We carry these phones with us. They have so much information, but how do you really pull these things in to advance our understanding of our environment? And then we're going to have a whole bunch of data. So Chirag Patel is leading this with Marina Sirota at UCSF. Chirag and Marina were graduate students together with Atul Butte at Stanford, who's one of the giants in this sort of area. And the idea is like figuring out how to do all this data analysis. How do you get these things together in a way where the data are comparable across the different studies, and what sort of infrastructures we need to actually have that sort of cross comparison there. And so this will be a really critical part where this is where we've been talking a lot, like to Google and AWS, is that we can't do all these things on our own. We need to have some of our partners, and thinking about how we can leverage some of these existing programs to build these things out. So I think this will be a very critical part to make sense of all this data we're generating. So this is the website that was just launched. We'll have things about content, about educational things. Ultimately, we hope this will become like a portal for accessing data sets and doing analysis. And that's something that you will see coming along, hopefully in the not too distant future. But we will be promoting a lot of different activities. And so if you have involved in any sort of Exposome related events, we will promote them on our website. One example here is this Exposome Moonshot meeting at Johns Hopkins in May. Hopkins has been very interested in Exposomics. They proposed this forum. They applied for an internal grant at Hopkins, which they got, which gave them money for the meeting and this entire building in DC to hold the meeting in. Like, so that was a great deal. I'm like, well, if they have that, we might as well work with them because they had that. And so this is going to be like, I think, a larger meeting that really looks at how to drive some of this Exposome work forward. And then just a couple of weeks ago, I was in Europe meeting with my European counterparts. IHIN is the International Human Exposome Network across the entire European Union. And the idea is we're trying to pull together that IRENE is a S-free infrastructure. These are European infrastructure things. We do not have a counterpart in the US for this. What they do in Europe is they build like 20-year programs around key areas that just get massive investment. So the CERN Collider is one of these projects. Some of the Galaxy tools that people use are part of that. BBMRI, all their biobanks come together. But this S3 project was recently funded to focus on environmental health in the exposome. And so we're coordinating with these other groups to really kind of form this global consortium with partners in South Africa, in Africa and South America, and Asia, to kind of pull these things together to really see how we drive the field forward. So with that, I just want to acknowledge all the people who have helped me do these things. If you want a good explanation of exposomics on a simple level, the Mayo Clinic did a podcast, their Future Medicine podcast, where Kathy Wurzer, who's an NPR reporter, interviewed Kostas Lazaridis and me. And she just asked really good questions, and they edited it down too. It was like really good editing they did. But I think there's a lot of opportunities to learn more about this. As Gata mentioned, we have a new journal for Exposome. So if you have Exposome related work, whether it be geospatial or more molecular, we're open to all things Exposome. And with that, I'd be happy to take any questions. Yes.
exhausted just listening to everything you're doing. So I have a question about the integration of the more laboratory measurements with the social determinants. And, a lot of us do stuff on social determinants, and particularly if you're looking at outcomes across aging. How do you grapple with the lack of specificity and the same level of precision in the social determinants? And I'm wondering what people are thinking about how to create mechanisms to match what you're doing in the lab.
So I think one of these things is, when I talk about how I approach the exposome, I realize that I'm coming from my particular angle. And this has been a place where working a lot with people that do more of the sort of social construct pieces is that there's things you can't molecularize, right? and there's some resistance to some of that. But at the same time, a lot of those social factors, like, for example, increase allostatic load and things, and these are measurable things we can do. And so what we've been trying to do is get more people on that sort of social side to engage to see how we can help, for example, what are the epigenetic signatures of this, early life trauma or something. And that's what I was so excited about that cancer study, was that we had a partner that was doing those really good social measures at the same time. And that's usually the problem, is that you have blood samples, but you don't have really good social characterization. And the idea of getting more studies that actually do that, I think will help show how some of those social factors increase vulnerability to multiple diseases. But again, there has to be biology behind that. It has to get transduced into some immune system change or something else in there. But if you don't have the right data, you can't make that comparison. So, this is where I think, for example, in larger cohort studies that have very good characterization of the social part, if you can just get biological samples from those people, you can start doing that. And I think it's getting more people to think about what is the value of getting that biological sample, and what are they willing to get? Maybe they can't get blood, maybe they're going to get a saliva sample or some other interstitial fluid or something. But I think it's getting the right people together to think about these problems and how they could work together to solve them. Yes.
Thank you. I assume that you, in some sense, want to understand causes. Is that right? With this,
To understand causes. Yeah.
To confounding and selection bias. How are you dealing with that in this study?
Yeah. So again, what my lab focuses on is measuring things in samples. So it's often the people that bring that study in that are looking at these things. I talked a lot to Mark Weiskopf at Harvard about these things. And one of the things that he's been really excited by is that when you can connect the geospatial piece and you have things in the environment, they become kind of anchors because they're not subject to reverse causation sort of things. And so when you look at your causal diagrams, if you have the right study design and the right connections to it, and have longitudinal samples and things, that you can address causation. But to me, I think what a lot of things that happen is that if you were to go to the doctor today, I won't be able to track out your entire history, but you are the summation of all the things that happened at that time. Whereas it might be more valuable in thinking about disease progression and response to therapy. So it's not always about the causation of the disease itself, but what you do with the person who has that disease now. So if someone's exposome, if you say that measurement means they're not going to respond as well to that drug, and you put them on a different drug and they respond better, I'm not as concerned about the causal connection of it because I actually was able to solve this problem there. So I think it depends. If you're looking at it from an epidemiological causal framework, that you have to do some things differently to do that. And the groups do that. But I'm also trying to help someone who has a multi-omic analysis study of a disease get more information on the environment. Can they address causation? Depends on their study design. Well, if you think about causal things with GWAS, for example, you're measuring genes that are fairly static, that there's a big advantage to doing that. But it still depends, like you have to have the right phenotypic characterization to see what you're going to see there. Like imagine like if you're thinking about doing like regular clinical chemistry for any condition, and you're looking at causation and you have cholesterol and glucose levels and things like that, we're just measuring thousands of things. It's still from a blood sample, like when was that blood sample taken and what things could confound it. And so again, it really kind of ties into what the goal of the different studies are. Like a lot of groups in Europe are using this from more of a biomonitoring standpoint. They're saying, what are people being exposed to, and just trying to regulate chemical exposure. So they're less concerned about causation because they know that chemical is already a carcinogen or something. So they just want to monitor it. So there's just so many applications to it, but we're just measuring these things, and it just gets down to what are those questions you're trying to address. And we have worked with a lot of people in the sort of causal inference area about what are those studies we need to do that. But it all comes down to what is that question you're looking at.
That's all of it.
Mhm.
So it's a way of leveraging all this to advance. How do you see different strategies to make that? That you look a little bit into the different active and different countries.
Yeah, I mean, so one of the advantages, and it's like all of us, is they've collected the blood samples and they're storing them for you, so you could come back later. They just started a pilot study of diabetes, about 5,000 people. And Jeff Ginsburg, the medical director of all of us, was showing me the data of type 2 diabetes and the incidence of it. And when they have 500,000 people in the study now, is they have people that were diagnosed a week after enrollment, 2 weeks after enrollment, 3 weeks after enrollment, all the way up to 3 or 4 years. Like the idea, because of that volume they have, and so if you're thinking about what's changing between that, they're collecting so much data from people that don't have that disease at time zero and are developing it over time. So I think that this diabetes pilot is a good one that they're trying to do now to see like what do they see in there, and can they identify the determinants of progression and onset and things. And so, I think that we know from UK Biobank and things, if you could do large scale proteomics, you can do it on the entire population, and there's all sorts of things you can do to assess what's going on. And I think we just want to have that same layer of an omic that covers more of those complex environmental exposures. Yes.
I have a question from our online community. The question is, do you think the concept of measuring the exposome could support the development of a more realistic demographic classification system to replace race by considering the environmental, social, and biological factors that shape and that shape health and identity, forgive me, and perhaps even replace race-based classifications in assessing and predicting health outcomes more accurately?
It's a good question. There's a lot of things that are kind of above my abilities to address on the race part of it. The one thing that we definitely know is that the majority of health disparities can't be explained by genetics, right? They're non-genetic, and that's what we're trying to capture with the exposome. And so if you're trying to figure out like what is the contributors to those disparities, I think exposomics definitely contributes to that. This is something that Melissa Davis, who's leading the Sandby project, she looks at the ancestry things, like that's what she's trying to get at in that study. So I think that it needs to be conducted with many other types of scientists to address that. But as far as like the disparities in what we're exposed to, we are measuring that. And like, so for example, like during COVID, if you looked at the areas in New York City that had the worst outcomes, like it wasn't their genome, right? It was something about their environment. It was very clear, like an environmental, it could have been like their access to care. It could have been, air pollution levels. It could have been all sorts of different things in there, but it wasn't their genes. And it was something in their exposome in some way. And I think that's how you get to those disparities, is by actually systematically measuring them. Yes.
Examples can you get one just across from the machine?
So, so the details here. So right now, our current rate is about $170 per sample. And we run like 80 at a time, like in a day. That's like what we put into an NIH grant. We do a lot of pilot sort of funding where there's someone who has 40 samples they want to do and they're writing a grant, and we just figure out a way to do it for them in that way. It does, one of the big goals of our ARPA-H grant is like we do LC and GC, so it's $350 if you're doing both of those, is to drive that down like under $50. Like just with more robotics, automation, we really want to drive those things forward. So we have to get that price point down. It's still not a bad price compared to other sort of analyses we do, but to scale, we need to get that lower. And so like I would say that, you know, from like, God and I have talked about these things from a collaborative standpoint. Like if there was a group saying, oh yeah, we're interested in this collaborative project, like we could do it collaboratively. Our lab's not set up to make money, but we got to pay the bills sort of thing. And so having collaborations and pilot grant sort of things, like we have those mechanisms to help people get started, to help them write those grants that can help get them funded in a longer term way. Yes.
How is that?
So yeah, mental state. I have postdoctoral training in neuroscience, so I actually think about some of these things quite a bit and study dopamine a lot. We have done some work on exposomics in collaboration with Yakov Stern at Columbia, where he was doing functional MRI and doing cognitive testing while they're doing the functional MRI. And so like that's something where like if you can study those levels of brain, you know, function and looking at these linkages there's ways of getting into that point. But when you think about like a functional consequence, now I'm thinking about more like homeostatic level things, right? So you think about if there's a social stressor or something to a person. And that's where I think that one of the connections there is we can isolate extracellular vesicles from the brain from plasma. And so we've been doing this recently where we can get it from multiple organs. And the idea is like, people did this for microRNAs for a long time, but we can actually do the same sort of mass spectrometry on it. So my sense is that when you have those sort of stressors and you see cortisol changes, there have to be other biochemical changes that are measurable in that, or an epigenetic change or something like that. So I think that gets into the sort of how are you measuring those stresses and allostatic load and cortisol levels when you also have this deep analysis at the same time. So I do think it's also the same thing, like having the people that collect that information correctly as collaborators to be able to do that. But I definitely think it's, you know, people in the neuroscience area study these things in many different ways, like from a brain imaging and cognitive testing sort of things. But it's just a matter of linking those things into exposures to tie it together. So I think it's possible. It's just going to, again, different approaches and designs are needed.
Okay.
So.
Okay.
Good points. All the differences. What are you kind of imagining outside of a drug state could be used for? What is the next need after that's done? And what's the risk to bring it in?
Yeah, I think that there'll be different conditions that'll benefit more from this, but we have to do the large scale studies to figure that out. Is if we can do, you know, full exposome-wide association studies, we will find out that certain exposures increase your incidence of certain different diseases. They make them progress faster. They make you not respond as well. Is the solution a policy level thing, or is it something the individual can do? It depends on where those exposures are coming from. But the idea is you'll have that data collected and not just like worrying about microplastics is important to do, but why just worry about that and ignore heavy metals? Like the idea, we want to look at all these things at once to prioritize, like where do you get the best bang out of the buck if you're saying it's this industry causing these problems for 14 different diseases, if we can head that off. Or it may be something where, like, we'll test for lead in a child that's having cognitive problems, but not just lead, right? I mean, there's so many other things that affect brain development and things. And so we have to kind of do that discovery piece to figure out which are those next steps. But I think that just like we did with like genome-wide association studies, you start seeing those connections, what combinations of genes increase that risk of the disease. What's nice about the exposome is that most of those things are much more modifiable than our genes are. And again, whether it be individual, policy, intervention, whatever it would be, is that there's more levers to move on the exposome side.